We've all seen images of crime scene investigators diligently solving crimes in television dramas. It all seems very glamorous, and in TV at least, they always get the criminal. But have you ever wondered if it's like that in real life? In this episode, we're going to find out by looking at some of the cutting-edge research being performed here in New Zealand in forensic science. Forensic science isn't just great material for a TV series. It plays an increasingly vital role in our judicial system, helping to provide a much clearer picture of a crime, how it was committed, and by whom. But what is forensic science? Answering that question is Dr. Michael Taylor, a scientist at ESR in Christchurch, one of New Zealand's most renowned forensic experts.
Forensic science is the application of scientific methods to the investigation of crime, and whether it be the presence of forensic scientists at a crime scene or back in the laboratory doing analysis on samples that might have been collected during the course of that investigation. It's essentially the same thing, the application of science to a criminal investigation.
The analysis of a bloodstain pattern is Michael's area of expertise. One of his current research projects is trying to understand exactly what happens when someone is shot in the head. The nub of Michael's research is in understanding the phenomenon of backspatter, material that is ejected from a gunshot wound. This matter can lodge on the clothing or skin of the person who fired the gun and can be a crucial piece of evidence in a trial.
Sometimes the court has to decide between an accidental shooting, a suicidal shooting, or a homicidal shooting, and the location of individuals that might be involved or thought to be involved can be quite crucial in making that kind of decision.
The theory is that backspatter material is ejected from the wound due to physical forces resulting from the bullet's trajectory through the head. As the bullet travels, it creates what's known as a temporary cavity, a wedge-shaped hole that closes up rapidly behind the bullet. Working on the project with him is Nardia Foote, a University of Auckland master's student.
There's an assumption that the collapse of the temporary cavity actually produces backspatter, but no research to date has actually proven that. There's a gap in the literature, so it's good to actually research it and determine if it is associated.
To test their hypothesis, they need to create a realistic model of a human head and undertake ballistic tests on it. But that's easier said than done. Finding materials that will mimic real human tissue and bone is very difficult. To begin with, they are creating simple models and using semi-opaque gelatin to represent the brain. These are then taken to a firing range and bullets are fired into them. On this particular day, they fired both 9mm and .22 calibre bullets into the models. A high-speed camera captured images of the bullets as they passed through the models.
By the time you hear the bang, it's all over. So the high-speed camera captures what happens when the bullet enters and travels through the gelatine and then exits. And you can freeze frame it and look at it just piece by piece by piece.
The slow motion footage clearly demonstrated some of the dynamics of their hypothesis. The formation and collapse of a temporary cavity as the bullet moved through the gelatin.
So you can see inside we've got the permanent cavity which is being left over from the bullet penetrating. The bullet passed through this end and exited out this side. So in terms of seeing backspatter, the backspatter would have come out this end. The hole is definitely bigger than the actual bullet, so it gives you an idea of the force that the bullet's generated as it's passed through.
Do you expect the amount of backspatter to be dependent on the size of the calibre?
It's very variable. Even in real life cases, you can't say if you use this gun with this bullet, you'll always end up with this result.
An important component of the research is trying to understand all of the physical forces that are occurring. To get a handle on that, the video footage is modelled in a computer so each frame can be analysed and measured. For this, Michael and Nardia turned to Peter Davidson, a senior research fellow at the University of Otago.
The real challenge is understanding what mechanical principles are coming into play, like momentum, energy exchange, and all that. That's a challenge. And once you understand that, then you can start applying equations and developing the mathematical model and see if you can explain most of the effects that you're after.
His analysis has shown that as the bullet travels through the gel, the temporary cavity oscillates and then collapses. The oscillation is due to the transference of kinetic energy from the bullet into the tissue. The tissue stretches and builds up potential energy. This rebounds back into kinetic energy. This recurs over and over, but diminishes and finally stops. This process, which takes milliseconds, is believed to cause the backspatter. So far, however, the computer modelling has not explained the process fully.
It doesn't explain the fact that the cavity tends to close behind it. And we believe the explanation for that is that we haven't modeled the air. We also need to model the air as well, because it hasn't, there's air rushing in behind the bullet.
The next step is to take what I've learnt from these pilot experiments and take it to the next level. So determine, are these simulants actually working in a way that is going to give us good data? The final model, not only do you want it to be anatomically correct, but the materials used need to mimic as closely as possible the human skull and brain and tissue.
Michael and Nardia still have a long way to go before they finalise their model. But this obviously challenging work is another important step in bringing scientific rigour to the field of forensic biology. Like any scientific field, it is continuing research which improves and refines forensic science. And here in New Zealand, we have some very smart postgraduate students who want to make their own contribution to this field of discovery. And I'm off to meet some now. First up is Ashley Fox, a master's student at the University of Auckland studying forensic science in conjunction with ESR. Her research project concerns fingerprints and the chemicals known as reagents that are used to enhance them at a crime scene.
Most of the fingerprints that you come across at crime scenes, they're not clear or they're partial, and you might want to enhance them a bit more. So the aim of the reagents and the chemicals is to enhance the contrast between the print and the surface so that you can see the ridge detail better.
Ashley wants to know if any of these reagents can interfere with DNA evidence that might be with the fingerprint. Her particular focus is on fingerprints made with blood. Knowing if this is the case could influence how evidence is assessed at a crime scene and help decide which reagent is best to use to avoid losing any genetic material.
There's always a toss-up as to whether the DNA evidence is going to be more important than the fingerprint evidence, and that's based on, statistically, is it going to identify a person? Fingerprint evidence is often quite a lot more identifying than the DNA, so they might get first pick. But if we know that we can still get the DNA and mRNA evidence after they've had their turn, then that would really, really help our evidence gathering in general.
At this stage, Ashley is trying to refine her methodology. She creates blood-stained fingerprints on glass slides. She can then test different reagents on them and see if she can still obtain genetic material from the sample.
I'm going to prick one of your fingers to get some blood flowing.
All right.
And I'm going to pipette a 5 microliter sample of that and spread it on another finger and get you to make some fingerprints on my slides.
Okay, so I need a finger with a lot of blood.
Yes, please. Okay, I'll just wipe that one down. So we know we're going to get the blood and no other contamination. You might have oils or something on there.
Right. Does this hurt you when you do it?
The pricking?
Yeah.
No, not at all.
Okay. I trust you.
I've never done it to someone else though. Did that work?
Yeah, there's a hole in there, but there's a bit of blood.
Just give it a squeeze so we get some blood.
Ashley takes a sample of blood, then smears it on one of my fingers. I then make a series of print marks on glass slides. It's on these she will test the reagents. Even though some of the print marks can't be seen with the naked eye, the reagents can make them visible.
If we were at a scene, that kind of fingerprint might not be useful for fingerprint officers, but we might take that one for the DNA part of it.
Right.
But if we're at a scene and this is all we've got, then that's when you have to start deciding
which one's
more
better.
important. Yeah.
So you could be changing the way that crime scenes are analysed in the future.
It will mean it's easier to make a decision when you're faced with that sort of fingerprint in blood. For the officer in charge of the scene, they can say, oh, well, I know that we're still going to get the DNA and RNA after fingerprint treatment, so we'll go fingerprints first. If my results show that we're not going to get the DNA and RNA results, then the officers will know that if the DNA RNA part is more important, then do that first, because any fingerprint treatment is going to ruin all your chances.
Ashley's research project has the potential to enhance the way investigators approach one of their oldest forensic investigation methods, fingerprint analysis.
One of Ashley's fellow students is also trying to create a new forensic process.
She is Elektra Williams, a master's student at the University of Auckland, and she is looking at a new way of trying to establish the post-mortem interval, the length of time from a person's death, in cases where a body is found outdoors. She is trying to extract genetic material from human finger and toenails that have been exposed to the elements to see if it degrades over time. Her hypothesis is that the more the DNA and RNA is degraded, the longer the person has been deceased.
Nails are quite resistant to decay because they've got a really strong biochemical structure that sort of could potentially protect the DNA and RNA inside the cell. So by using nails, it might not be so influenced by environmental variables and like body acids as things like soft tissue can be.
Her experiment involves putting the nails in various environments, such as in soil or water, to see how they degrade, and then seeing if she can get a sample of genetic material from the nails. The end result would be to see how much the DNA or RNA is affected by those environments.
What I've got to do first is get a sample of your nail clippings that I can work with. So I've got some nail clippers you can use here.
Yeah.
Try and make it so the nail's not completely fragmented, like as big as possible.
As big as possible.
The next step is to put the nails in different environments for a period of time. The aim is to try and simulate a body lying in a real outdoor location.
So what I'll do next is I've got two different mini environments set up. So here I've got a tray of soil that I just grabbed from outside, and I've also got a small beaker of tap water, just so that I can do a comparison between the two.
After the allocated time, she will recover the nail clippings and then clean them to remove any contamination. She then puts them in a chemical solution called a lysis buffer, which breaks down keratin, the protein that gives nails their hardness and strength. The lysis buffer is incubated for 40 minutes, and the resulting solution then contains both DNA and RNA. She takes the RNA, amplifies it, and then can analyze it. So far, she has managed to identify 3 types of keratin RNA. This is an important discovery as she needs to be able to prove the sample is from a nail and not any other type of tissue.
Later on in the year, I'm going to be setting up a more long-term, bigger scale environmental experiment where we'll be having like more fingernails and toenails in lots of different environments where you might find a body in New Zealand and with a longer time frame. So I might do that up to 4 months. So I'm just trying to get a feel of what the results might be like.
If Elektra can perfect her methodology, she may have created a technique which will show how long it takes for RNA to degrade when a nail is exposed to the elements. Getting a handle on that may help determine how long a body has been in that location.
It's very early days for Elektra's research, but her initial success could lead to another forensic tool for crime scene investigators.
Up next, we meet a researcher who is studying the decomposition processes of bodies found in water.
Whenever a dead body is found, there's some major questions that the investigators need to answer. What caused the person's death? How did the body come to be in the location it was found? And how long has it been there?
We're about to meet a researcher who is doing a unique and rare piece of research, trying to determine how long a body has been in the ocean and what steps are involved in marine decomposition. Gemma Dixon is a PhD student studying at the University of Otago. She is particularly interested in how marine bacteria are involved in the decomposition of human and animal remains in coastal marine environments. She believes that the bacteria could act as a post-mortem clock. Waves of bacterial colonization could indicate submersion length.
It's important because at the moment we don't have a scientific method to determine how long a body has actually been in the water for in a forensic context.
To find out how bacteria colonize a body in water, she places pig heads in a cage and submerges them in Otago Harbour. She uses pigs because they have similar skin structure to humans and decompose in similar ways. From this matter, she can take swabs over a period of time to get samples of the bacteria and then identify them using DNA analysis. To see how she does this, I went with her to Portobello Marine Lab.
We're going to swab in 3 places, the snout and the cheek and the neck wound site. Basically we're looking to see if there are any differences in colonising bacteria in these 3 locations.
Why do you swab in those 3 places?
Because if we had a human body, we would ask the question, does it matter where we swab on that body? If you had a whole body or say a leg with exposed muscular tissue, we want to know if there's any difference between these sites. So does it matter where you take your sample from?
Okay, so maybe a wound or something as well.
Yeah, exactly.
Yeah. Cool. Should we give them a swab?
Swab. Okay, so I've labelled these already. This is wound. So basically just take a good sort of 2 centimetre square
I guess
swab.
That's the good thing about working with bacteria is you only need a small amount of area.
Yeah. There's generally quite a lot of bacteria on there.
So will there be a bit of bacteria on them now?
Yeah. They do have bacteria on them to begin with. And I like to know what was there before I put them in the water, so I can see the changes that occur once it goes into the water and over time.
Once she's taken the initial swabs, she places it in a cage and then takes it to a spot on the wharf. It's then lowered into the sea. For the first 3 days, she swabs it daily, and then every 2nd and 4th day until just the skull remains. This usually takes about a month.
So how can you be sure how much has been taken off by bacteria and how much has been taken off by fish?
It doesn't matter too much.
Right.
I'm interested in the bacteria that are colonised more than the tissue that's been removed. Decomposition is quite a messy process. It'll get messier and messier, and then gradually you get tissue loss and the smell dies down as well with the loss of the tissue. So it's not too bad. When I'm swabbing, I just stand upwind of it and you don't notice the smell so much.
While decomposition on land and in the sea have some similarities in terms of being colonised by different organisms, there are notable differences. The most notable is that on land, insects play a major role in decomposition.
What happens in water is not necessarily translatable to what happens on land because there's different bacterial groups involved. In water, decomposition generally happens much slower, and that's generally because of the colder temperatures of the water. Yeah, temperature plays a huge role in the progression and speed of decomposition.
In doing this work, Gemma has been able to prove that bacteria do colonise mammalian remains in the sea, and this colonisation occurs in waves of succession.
There are some that colonise early and you don't see them later on. There are some that colonise during the mid stages of decomposition when there's a lot of things going on, it's really messy. And there are some that colonise later, after these other ones have come and gone. So they seem to all have their particular role in the process, and they come and go at different stages.
Through her research, Gemma has made some world-first discoveries.
I've also found several bacteria that are new to science. I found them based on the sequence of their DNA, so there's nothing really similar to them that have been found before. And these ones actually seem to be quite important in the process as well. I found them in quite large numbers, so they're particularly interesting to us in terms of their role in the decomposition process.
Gemma's research project is truly unique. The next steps would be to assess seasonal and geographical differences and see what effect clothing would have on the decomposition process. And though pigs are seen as good approximations for humans in experiments, she believes that at some point human tests would be needed. That said, she believes she has begun what could be a promising new line of investigation.
I think with more research in the future that this could actually be used as a more scientific and reliable tool to time how long a body has been in the water. That would be the ultimate goal, that it would be used in a forensic context, but it would need to be well studied before it ever got to that point.
Forensic examinations are an indispensable part of any crime scene investigation. The work is hard and involves detailed recording and analysis of specimens and data.
Thanks to dedicated researchers here in New Zealand, we are getting a more and more precise understanding of forensic biology, which is giving us greater confidence in the evidence taken from crime scene investigations.